Okay, so it's 3 o'clock British summertime. I'll just begin. I think more people are joining at this moment, so I am just going to do my introduction and then we'll go from there. So good morning, afternoon and evening to everyone here, depending on where you are today. My name is Jonathan Best and I am the Research Director at Cell Guidance Systems. It gives me very great pleasure today to introduce the launch of our new combination product, PODS-PeptiGels, which has resulted from an exciting collaboration between Cell Guidance Systems and Manchester Biogels. For those of you unfamiliar with us, Cell Guidance Systems is based at the Babraham Research Campus in Cambridge, UK, and provides cellular reagents and molecular tools to characterize and understand protein delivery systems. Natasha Lewis, our biomaterials scientist, will be presenting the PODS element of our portfolio. Manchester Biogels is located in Audley Park in Cheshire and is a leader in the design and manufacture of peptide hydrogels, which Dammy Olayanju, principal scientist, will kick off the presentation with in more detail. Together, they will explain how this combination product can enhance and help your cell culture needs. The presentation is going to take around 25 minutes and we will have time for questions and answers at the end. So if you'd like to submit your questions to the answer and question bar at any point, we'll go through them then. For everyone's information, this event is being recorded. So if your colleagues are unable to attend, we'll be making it available after today. So without further ado, I will hand over to the presentation with Dammy and Natasha. Thank you very much.
All right, thanks Jonathan for introducing Natasha and I'm very excited to actually explain or take you through the field of 3D cell culture and how we've come to the discovery of PODS-PeptiGel, which will be the main focus of today's presentation. So just to give you a brief background of why PODS-PeptiGels have actually been discovered. So when you look at the 3D cell culture, I'll talk about the challenges. When traditionally looking at 2D culture versus 3D cell culture, there's a big drive coming from 2D culture into 3D culture. 2D cell culture has been used for a lot of studies over the years, but it's got a lot of limitations. So 3D cell culture system in particular has been used, to better mimic what you see within the in vivo environment. One, because you get to see enhanced cell-to-cell interaction and signaling. And also it helps to have reduction in animal usage, which means they're compliant with the 3 Rs. It also allows us to have that complex 3D systems, things like integration of fluid flow and bioprinting. And taking this all together leads to us having a reliable and relevant result. So when you look at 3D cell culture also, because based on all this taken together means you can have physiologically and clinically relevant results. And you can then understand why people are moving from 2D cell culture to 3D cell culture. So within 3D cell culture, there are 3 main key challenges that most scientists have been trying to develop and address over the years. This is looking at cell biocompatibility, scaffold tunability, and functionality. So an understanding of these 3, or the optimization, let me say, of these 3 aspects together will help us to have that fully developed cellular phenotype. So I will just focus slightly on scaffold tunability, choosing the most suitable type. So in terms of the scaffold tunability, or choosing the right scaffold for your cell type broadly, there have been 2 classifications where you can either have the natural scaffolds, just looking at things like proteins, polysaccharides. On the other hand, you can have things like synthetic scaffolds, looking at things like polymers, ceramics, and metals. So when we look at the natural scaffolds, these got advantages because they are functional and they are biocompatible. But then when you look at the disadvantages, because they are natural, coming from more of living organisms, you have things like batch-to-batch variability. They are not defined, they are not scalable, not translatable, and they have poor mechanical strength. And all this, even though they can be used to create 3D structures, looking at the disadvantages means you can't fully translate your in vitro results towards clinical interpretation. On the other hand, you can have things like synthetic scaffolds, which they are ethically compliant, they're fully defined, scalable, there's no batch-to-batch variability. On the other hand, you have things like limited biocompatibility, they are not functional, and most times they may require chemicals for stimulation. So all this we've taken together from, taking the USPs of natural scaffolds plus the USPs of synthetic scaffold in terms of the advantages, pull them together. And this is what we do as a company from Manchester Biogel to create what we call PeptiGel, which are peptide-based scaffolds. And I'll come to explain that as we move on towards the presentation. So in terms of functionality, there are also issues with, the growth factors. And then, talking about 3D cell culture, if, from the previous slide, talk about the cell biocompatibility. So firstly, when you have the scaffold, the cells need to like the environment, they need to interact. You have cell-to-cell interaction, you have cell-to-ECM interaction. And once you're able to establish that nice cell-to-ECM interaction, you need the growth factors to be able to create the signals for various signaling pathways for cells to be able to function accordingly. But then, traditionally, a lot of growth factors have relied heavily on the use of recombinant growth factors. But the issues with this is they degrade quickly, and there's a need for constant replenishment. Now, if you look at that aspect on its own, because cells, they go through different stages, even when you look at the cell cycle, when they're going through different signaling pathways, there's the need for constant replenishment of growth factors to be able to maintain that signaling pathways. And all this leads to system complexity. So which means there's a lot of work that needs to be done for, one to really understand where the probably the timings of, let me say, growth factor replenishment is really required for optimal cellular behavior. Taking all this then leads to cell stress, and with a lot of different products in markets, there's product variability, and altogether this leads to high cost and may not be the most optimal choice of growth factors for having that best cellular phenotype that one really wants. So this aspect is going to be explained further by my colleague here, Natasha. So lastly, talking about cell biocompatibility with scaffold, this is looking at the interaction of cells with a scaffold for optimal cellular phenotype. Now we've talked about the scaffold, we've talked about the use of the growth factors. Now, all this, is required to really have the optimal cellular phenotype. Now, when you look at the way cells interact with the scaffolds, largely you can look at the physical chemical properties of a particular scaffold will determine how a cell responds. So the optimal combination of the size of the scaffold, the shape, composition, the degradation rate, even the material chemistry, surface wettability, roughness, charge, hydrophobic interactions, competitive protein binding. The mixture of all this is actually very complex, but understanding all this is really, really required for the cells to be able to behave in the best way as possible. And this is where, with the understanding of this, understanding of the role of the growth factors, understanding the role of the scaffold is what's actually led to the new product we're introducing to you today. Now, when we talk about the platform technology, looking at the scaffold, now the 3D scaffold, the PeptiGel, which is what we produce as a company from Manchester Biogel. Now, this is taking, making use of synthetic peptides, using the natural building blocks of any natural tissue. So you have this synthetic oligopeptides using a lot of defined protocols, things that have been heavily invested over the years as well. So we've been able to change or optimize the parameters of different amino acids in terms of the pH, the time, the temperature, functionalization. And when all this is done in-house from our IP technology, it leads to a nice self-assembling of these peptides to form nice beta sheet structure. So this all together, you have the fiber entanglement when you have the solvent creates the nice 3D environment for cells to actually grow and thrive in. And that's why, by the time you have the 3D nanofibrous hydrogel, which comes ready to use, they can seed your cells to create that nice optimal environment for your cells need. So as an example, just to show an image of fibroblasts growing in this PeptiGel technology, so you can see that cells love the gel and they can proliferate in the gel, as shown from the live and dead images of this particular cells grown in PeptiGel. And just to finish up, talk about the advantages and the applications of the PeptiGel system. So in terms of the USPs, they come ready to use with a lot of traditional scaffolds and markers. There's several steps required. Some you may need to defrost on ice, as an example, and it means your gels come ready to use, and you can just seed in your cells and off you go. They are biocompatible and translational, so which means what you develop in vitro can be rapidly accelerated into the clinic without not thinking so much about the regulatory aspects of if a particular product can be used in the clinic. They are injectable, sprayable, and printable. Because of the shear thinning properties of the gel, it means this can be used for other aspects of therapeutic applications in terms of cell therapy, where you can spray onto wound bends. The animal-free and ethical, there's that big drive towards reducing animal usage in research. So because they are chemically defined, it means you don't have to worry about animal usage. And because they're also chemically defined, the gels can give reproducible results because there's less batch-to-batch variability. They are transparent. It means you can image yourself, as we've seen previously in the slide showing the growth of fibroblasts in the gel. You can fine-tune the other parameters of the gel in terms of the mechanical strength as well. So taking all this together helps us to be able to create that fine, optimal environment for a particular cell type. And this can be useful, as we look into the application areas of PeptiGel. So in terms of using PeptiGels for different applications, they'll be used largely for creating different complex 3D models, things like spheroids, organoids. They will also be used for, creating, co-culture models, coating of plates for different applications as well. So it means it's not, it has a lot of wide applications with usage. And because you can really change the parameters of the gel, they're nice to be used for disease modeling. They can be used for drug discovery and drug screen applications as well. And because of the shear thinning properties, also they can be used for bioprinting applications. So which means you can create, your cells within the gel nicely, and also by the time you want to think about having complex structures in terms of bioprinting, the same gel can be used in that aspects as well. Regenerative medicine, as an example, is where we've differentiated mesenchymal stem cell, for end-based cellular therapy kind of applications. They are also applicable for use in terms of liquid handling. So which means they can be used for high throughput application as well. And that's an example showing where we've had little droplets of the gel. So taking all this together means, in terms of the innovation of using PeptiGels, it can be used for a wide array of different preclinical and clinical based kind of studies. So as said earlier from my introduction, looking at the growth factors. So at this point, I'll hand over to my colleague Natasha, who will explain the growth factors and how we've actually come to the discovery of the PODS-PeptiGel to create that fine optimal environment for your cell needs.
Thank you, Dammy, for that great overview of PeptiGels. I'm the biomaterial scientist here at Cell Guided Systems, and I am now going to take you through our technology, PODS. So PODS stands for polyhedron delivery system. PODS consist of a an encapsulated cargo protein within a protein crystal shell. The cargo protein is normally a growth factor or a cytokine. In the presence of proteases, pores will form within this cubic crystal, and this enables the slow and sustained release of the growth factor cargo. Because of the crystal formulation of pods, they are very robust, they are temperature and pH stable, and therefore can protect the cargo from chemical and physical stresses. Very importantly, they are also animal-free. The sustained release of PODS cargo into the media in a cell culture system enables the spatial and temporal control of the cell culture environment, whilst significantly stabilizing growth factor concentrations to improve the cellular phenotype. There are many further benefits of PODS, including that they're biodegradable, non-inflammatory, and because they're a platform technology, we have many different PODS growth factors available. PODS protein crystals are based on a baculovirus construct, which is expressed within insect cells. So that construct contains both the polyhedron protein and the cargo protein of interest. Those proteins are co-expressed within the cell, and the cubic crystals naturally form, as you can see in the middle picture. They are very easy to purify. The cells are simply lysed and then centrifuged to remove any cell debris. I'm now going to show you some examples of how PODS can be used in cell culture. So these graphs show a concentration of a particular growth factor, GM-CSF, both originating from recombinant and PODS GM-CSF. So what I'd like to draw your attention to in these graphs is the y-axis. So both of these are in picograms per mil, and in both situations, the starting concentration is around 2000 picograms per mil. However, for the recombinant GM-CSF on the left, after 1 day, the levels have dropped drastically to 200 picograms per mil, and over a 7-day time period, those levels reduce even further. This is compared to the PODS graph on the right, which is levels over 30 days. Again, that starts at just over 2000 picograms per mil, but you can see that the decrease is much slower, and at 30 days, we actually have even more growth factor remaining in the system than at day 1 with the recombinant growth factor. So this is a really great example of how PODS can overcome some of the issues of growth factors that Dammy was talking about earlier. PODS can also be embedded in a range of different hydrogels, including PeptiGels. So these images are of PODS which have a GFP cargo for fluorescence imaging, and these are embedded in PeptiGels. On the left, you can see that the PODS have been evenly distributed throughout the gel. And then on the right, this is a 3D bioprinted structure with Pepti ink either with or without the PODS. And you can clearly see that they can be localized within the structure precisely. In this way, PODS can be used either dispersed throughout the gel to provide a homogeneous distribution of the growth factor, or they can be localized within a structure to set up a concentration gradient, or for patterning within a tissue model, for example. There are many different applications of PODS, including some of these listed here, and I'm sure there are many that I haven't thought of, but these include complex 3D models, for example, organoid culture, stem cell maintenance, microcarriers and biomanufacturing, and then biomedical and regenerative medicine. So having reviewed our PODS technology, that leads me on to talking about PODS-PeptiGels, which is our new combination product. So PODS-PeptiGels combine many of the advantages that Dammy and I have just talked about to produce a tunable scaffold which has a specific slow releasing growth factor, And these are biocompatible, cell-specific, and can be functionalized to any requirements. The method for using these PODS-PeptiGels is also really straightforward. So because PeptiGel and PODS are both shipped and stored at room temperature, it's simply a matter of mixing them both together with or without cells, adding the mixture into your experimental well plate, incubating as usual, and then analyzing as you would with any other hydrogel. As I mentioned previously, proteases are required for release of PODS cargo into the surrounding culture. And depending on the release kinetics of the growth factor that you're working with, you can tailor the combination of PeptiGel for your requirements. So these two graphs display data of PODS cargo released into the media surrounding different PeptiGels. On the left, we have release from PODS in alpha-2, and that's in both serum-free and serum-containing conditions. You can see that those release profiles over 14 days are very similar. So this means that with this gel, the PODS cargo is inherently released, and this would be suitable for use with cells that require a serum-free culture system. Then on the right, we have the same experiment except using alpha-4 this time. And in this scenario, exogenous proteases from the serum were required to give the desired level of growth factor release, whereas without serum, the release is very minimal. These two gels also have different properties themselves, but they differ both in stiffness and charge. And I think you can see that there are many parameters that you can control with PODS-PeptiGels to really tailor it towards your own requirements. So I'll just move on to speaking about some biological data that we have using PODS-PeptiGels. So in this experiment, we've used fibroblasts in PeptiGels, either without any growth factor with conventional FGF2 or with PODS FGF2. And in the graph on the left, you can see that the PODS FGF2 have produced a much higher proliferation compared to the two other controls. And this corresponds qualitatively to the microscopy images, which are shown on the left, where you can see that the cells are much more rounded in the two controls, and with the PODS, they have spread out and proliferated much more. So the sustained release from the PODS at the beginning of the experiment has maintained the health of the cells throughout the period and has increased proliferation. This is another example of how PODS-PeptiGels can be used with different cell types. So these are C2, C12 murine muscle cells, and these have been grown with both FGF2 and IGF1. Again, as in the previous experiment, the PODS perform better compared to the conventional growth factors with an increase of cell proliferation over the 10-day period. Again, there are many applications of PODS-PeptiGels. So just a few of those are bioprinting, neuronal differentiation, disease modeling, regenerative medicine, and drug discovery. But there are many more applications, and they could be used for any 3D cell culture. So the PODS-PeptiGel technology has its own set of benefits, and here we have comparisons to several different types of other hydrogels that are available. So including animal-derived, non-animal-derived, such as alginates and cellulose, synthetic materials, and hybrid materials. PeptiGels are compatible and non-toxic for all cells. They're very easy to use, and they produce reproducible results because of the synthetic peptide. They can be tuned to suit your cell type in terms of stiffness and charge. They can be printed, used in translational studies, and don't have any immune response. And the final line on this table shows that they have controlled and sustained release of growth factors, which isn't the case with any of the other products on the market. So now I'll move on to how to choose your PODS-PeptiGels for your application. So we have tried to make this process for choosing which pods and PeptiGel you need as simple as possible. So firstly, identify the growth factor you require, which will probably be similar to any conventional growth factor that you might be using. Then choose the PeptiGel for your cell type, and we have a recommended list for different types of cells, and then they will be shipped to you together. So we're starting off with 10 different pods available. That's 3 controls and 7 different growth factors. And these come in standard pack sizes of 50 to 150 million and 1 billion pods. And there are also 9 different PeptiGels to choose from in standard pack sizes. And these are, as I mentioned, can be suitable for different cell types. With the controls for the pods, we recommend purchasing these alongside pods which do contain growth factors. So the pods empty are just simply polyhedron crystals without any growth factor, and those are intended to be used as negative controls. Whereas the 2 fluorescent controls, GFP and M-Cherry, can be used as visualization tools to observe the location of different pods. All of these 3 do not elicit any effects on cells, and their purpose is to be negative controls. We're here to support you at every stage with the PODS-PeptiGels. So we have a range of online resources for further reading, which includes protocols, application notes, and guides. And if you have how to plan or set up your experiments, please feel free to get in touch with either myself or Dammy, and we are very happy to provide you with help and advice. So thank you very much for listening to our webinar today. Please feel free to get in contact, and we're happy to answer any questions that you might have.
Great. Thank you very much, Dammy and Natasha. That was very informative. So we have had some questions being put into the question and answer panel as we go through. I will read some out. I mean, we should have enough time to answer most of them, I think. But I'll start at the beginning from Christina. She'd just like to understand a bit more if the peptides that are used in the PeptiGels are derived from defined extracellular matrix protein, or they are random peptides. Could you explain a bit more on that, please?
Yeah, thanks, Jonathan, for that question. So just to take it backwards, to start with, the peptides are synthetic peptides, so they are not from animal-derived matrices. And secondly, this is using the natural peptides you have within the native tissue. Things like glutamic acid, just as an example. So you have a list of different peptides. So what we've done as a company is to try different combinations of these peptides to create the nanofibrous extracellular matrix structure that can allow the cells to thrive in there. And secondly, to make a more representative of the extracellular matrix within the in-vivo environment, then we can introduce biomimetic peptide sequences of different ECM proteins that you see naturally. And that's what we do as a company.
Great. Thank you very much, Dammy. There's another great question here, which from Gloria, who says, guess that's growth factors can enter the gel reaching the cells, but have you got any information about small RNA or DNA sequence and whether it's possible to transfect the cells in this gel system?
Yes, so it's possible to transfect the cells because when you look at DNA sequence, most times they're around 2.53 nanometers, and because the gel is porous, which allows the diffusion of growth factors, some of these proteins or growth factors are even larger than DNA sequences. So, if you want to transfect the cells, because you can have growth factors and different things get to the cell, then it's possible to have, your DNA sequences to transfect the cell.
Great, thank you. There's one question which saying, could you share the slides? And just to let everybody know, these will be available from the websites alongside the recording. So yes, the slides will be shared. Okay. So another great question, is it possible to create oriented nano microfibrous structures using PeptiGels?
Yes, so naturally when the peptides self-assemble, the end product actually is nanofibrous structures. So which means, so the answer is yes.
Okay. Great. Another question saying, who says, thank you for a great presentation. Sounds like a fantastic product. So thank you very much for that. I wondered if you could give your opinion on what the best combination you think for intestinal organoids. Is there a short answer for that, or is that one that we can follow up with later, do you think?
And so in terms of intestinal organoids, the first, well, in terms of the peptide selection or PeptiGel selection, we have different PeptiGels that we've seen proof of concept, and we are actively working within this as well, within colorectal organoids, where they've been shown to support the growth and propagation of intestinal organoids. Now, when you look at the media and understanding the stem cell differentiation pathways of organoids, there are certain growth factors that are also important. So in this case, things like FGFs, for example, EGF, where people have really optimized the structure, and all this, we have the pods available for those as well. So the main idea is when you have the PODS-PeptiGel, in this case for intestinal organoids, then we can have that nice customizable environment for your intestinal organoids. But we are happy to be able to schedule a call and advise better on this if that's something you're interested in looking at.
Great, thank you. So this is a great question coming up now from someone is, why should I choose your kits over Matrigel?
Okay, so I guess I'll take that one. So first of all, both of the components are completely animal free, so they're both synthetically made. There's no mice involved in the production of our products. They're also much more defined, so because you can choose the stiffness and charge of your particular gel, you can match it up to your cell type, and there'll be much less variation than compared to Matrigel, which is because it's biologically derived, there will inherently be a lot more variation. And the same with the growth factors, so you can control the amount of growth factors that are there. You can put in more than one if you need to, and you can also control the amount of pods that are put in there, so you can control the release of the growth factor over a period of time.
Great, thank you. So yeah, next one from Katerina, who says, nice presentation to both of you. She would like to know if you got other examples of cell types that we've had experience with. She said you've shown a nice examples of fibroblast cultures. What other cell types have you used in your experience with them, or even cell lines, in fact?
Yeah, so we've tested fibroblasts. We've also got some data with C2, C12s, which are murine muscle cells. We also have some experience of blood cell lines, and some collaborators have been looking at otic neuronal progenitors. So we have some nice data which we'll hope to present later in the year about how they can influence progenitor growth and neurites. But for both pods and the PeptiGels, we have a lot of examples where both our products have been used, and it would simply be a matter of using them together. But of course, we would be happy to have a call to discuss that further.
Great. I'm just going to answer some simple ones. There's one which says, great presentation. Are these available to buy now? And the answer is yes, they are. We've had the information from the website, which will be available both on this presentation and we will send out afterwards. So yes, please go ahead and visit the site and buy as many as you like. Somebody else said, could you, I believe you provide advice on the type of PeptiGel to use for particular cell lines. I mean, do you want to just outline how you would go through that process if people want to get in contact?
Yes, so as a company, we're very customer focused. So normally you send your inquiry either through the website or you can ring us as well. So we do schedule a call, just to understand what you're trying to do and to be in the best position to give you the best advice towards the type of PeptiGel to use for your cell type and your application.
Great. Sylvia says, are the growth factors synthetic?
So yes, they are synthetic. They're made using a genetic construct inside insect cells, and we just grow them up inside the cells and then purify the crystals after that.
Great. Jill also said about sharing PODS publications, and we can definitely share that. There will be, again, we have a number of publications on the Cell Guidance Systems websites about PODS and how they can use in their applications. So we'll send those links out after this webinar today. Lucas asks, is there any way to customize the rate of release of growth factors, or is it a standard release rate for all factors?
So there are a couple of ways that you can look at controlling the release rate of the growth factors. Some of the data that I presented was about how there's a different release rate depending on the PeptiGel that you're using. So that's one of the ways you could test a few different PeptiGels and see which ones have the desired release rate. And then the other way is to vary the concentration of serum that you put into the media. So because the mechanism for release rate of growth factor from PODS is through proteases, if you vary the amount of proteases that you're adding, that can also influence how quickly the growth factor is released. So there are a few different ways that you can do that.
Okay. Marie is asking about what is the concentration of total proteins in PeptiGels and what is the type of proteins inside? Are they growth factors in PeptiGels alone? Are there growth factors in PeptiGels alone?
Yeah, so I think there are three parts of the question. The first one, in terms of concentration. So unfortunately, I won't be able to share that because that's trade secret. And the type of protein inside, like that's talking about like a formulation. But the good news is obviously depending on the type of work, if there's a need to understand the formulation, then we can sign an NDA, just to be able to go through what the product contains, towards that particular application. In terms of growth factors, so there are two categories of PeptiGels. We have what we call the bare PeptiGels, where there's just pure peptides without growth factors. But based on the structure of the products, then you can have growth factors embedded in the PeptiGel, which also is what has led to the discovery of PODS-PeptiGel.
Great. So there's quite an interesting one here from Gloria. Are the PODS compatible with magnetic resonance imaging?
So I'm not sure of the answer to that question, but I think we might have some data from collaborators. If you'd like to get in contact with us, we can talk to you more about that.
Yeah, and I guess how they're being used as well is something that we could talk about further. A great question. Okay, Kosher has said, would using this gel be good if I'm testing the increase or decrease of extracellular matrix? Would there be any mix-up with ECMs?
Yes, you can. They can be used to test the increase or decrease based on the experimental setup. I guess what's coming to my mind is obviously when you have the cell setup, the cells will secrete extracellular matrix protein, and based on the endpoint analysis, we determine how you measure that. But we can schedule a call to actually understand what they're trying to do, and then to be able to give the best advice technically.
There's quite a few questions flooding in now, so so do you have any experience in generating brain tumor organoids derived from cancer stem cells alone and with neural cells? That may be a follow-up, but I think if you have any opinions on that one.
Yes, so we have some customers and collaborators looking at brain related systems, and just like you said, I think we'll be advisable to have a follow-up, and then we can have more technical call on
that. Somebody else saying, can antibodies enter these gels?
Yes.
Yes, they can.
Short
and sweet, nice one. So there's quite a lot of questions about combinations for tumor organoids. I wonder if we could probably sum up how, so a number of people are asking what sort of combinations would be good, and if you have experience with them. Maybe if you want to, if you can summarize, Dammy and Natasha, probably how best to think about when studying tumor organoids, and the best sort of either combination or how best to get in touch with you, and the kind of applications that you've seen.
So the first aspect is on identifying and understanding the key cellular pathways as relates to the growth factors. So once we identify the growth factors, let's say, for example, maybe transforming growth factors beta is really key to what they are trying to investigate, then we can then match the best PeptiGel or the one that's been optimized for that particular cell type. And once we've been able to come to that conclusion, then you can send the order in, and then you can get the right growth factor of interest and the PeptiGel that is suitable for that application. And then you can, and there's a protocol to guide the user in doing how to do this.
Right.
That would then be the best combination product to take that study forward.
So I'll just read this one because somebody's saying great suit, Dammy. I'm assuming that's somebody you know. I'm very jealous of it, of course. But it's I think that the question is, how many pods do I need, and how much PeptiGel do you recommend for 3D culture in a 24-well plate?
I guess at the risk of sounding a bit repetitive, we would encourage you to get in contact with us so that we can tailor the amount of gel and number of pods that you would need to your application.
Okay, that's great. Okay, there's one. Do you see any issue with autofluorescence?
No, and like Natasha showed previously, she hasn't shown like the full data where you had the PeptiGels on its own, and then when we had the GFP pods, which have been, pods that have been incorporated with GFP, are embedded in PeptiGels because the nice autofluorescence, or the fluorescence of the GFP pods, but the control to that had no fluorescence from the gel.
Right. So there are a couple of questions about the technologies being used in co-cultures. If you have, if you could answer that one, are they being used in co-culture scenarios?
Yes. So when you there's a study that's actually published, so we're happy to share the publication that's looking at Barrett's esophagus, where you have the non-healing of the esophagus, and what surgeons normally do traditionally is scrape up the esophagus. So to mimic that, in vitro was to do a co-culture where you have fibroblasts in 3D and then see the epithelial cells on top. And also these are extended to different wound healing, this kind of application, skin cultures where you have fibroblasts and have keratinocytes on top. And for very technical based kind of questions, we'll strongly advise to get in touch with you after to go through results and guide you best in designing the experiment.
So yeah, we've got a few quite specific questions around cultures. So I probably would, in the interest of time, because we've got quite a few more questions to go through, probably to contact Dammy and Natasha directly if you've got any questions about actually how it would work and if there are special situations. So I think you probably answered most of that. Yes, we do send a certificate of attendance for those who need them or are interested in them. We can certainly do that. So there's sort of some of the specific questions are, do you see the same release, presumably of the growth factors, when you use human platelet lysate rather than fetal calf serum or fetal bovine serum?
So I'm not sure if we've actually specifically tested human platelet lysate head to head, but we would encourage you to test before you go into any experiment to just see how your release rates and dynamics are affected by the particular serum that you're using.
Christine says, is it possible to have customized pods expressing a specific protein?
So the short answer is yes. We have a catalogue of PODS growth factors and cytokines. I think we have over 90 already in the catalogue. So you can go to our website and see if your protein of interest is already there. And of course, you can combine those with PeptiGels. We're very happy to do that. And if you have something that's not in the catalogue, then we could potentially also make you a customized growth factor, and you would just have to get in contact with us about that.
Yeah, that's great. So Katerina is planning on doing some long-term culturing experiments on organ chips in a microfluidic device. Do you have any experience with this?
Yes, so and the aspects of the technology, which because of time we didn't really talk on, is the bioprinting side. And this is what we refer to as PeptiInks, because the gels are shear tinning, and you can also fine tune the mechanical stiffness, so they can actually be used for organ on the chip, fluidics-based kind of applications where you can print little droplets. And also, if you, I don't know how much you remember based on the presentation, the slides where we're talking about the applications, there are little droplets that are printed on the plate. So all this can be used for organ on a chip or very, high throughput based kind of application.
Mm-hmm. And the particularly interesting thing about using pods with the PeptiInks or the PeptiGels is that you can localize them within your structure or microfluidic device, so you can set up a gradient of growth factor within there if that's of interest.
Okay, that's great. There's another question about microfluidic devices, which I think we've answered. Somebody's asking, have you tested the pods in other bioinks, and was the result as good as in your gel?
So we have tested pods in a few different inks. I would say that the PeptiInks were by far the easiest to work with, just because there's no temperature requirement. They're very easy to print with, and it's very plug and play. And we definitely got the best results using those Pepti inks. But it is possible to combine pods with other inks.
Great. I think that's why the collaboration actually ensued, because of the ease with which we found the combination product to use, wasn't it, really? So I think that it's not just, serendipitous, actually, it was worked through quite a lot. So have you been able to, Jonathan's asking, not me, another Jonathan, there's more than one of us, it appears. Have you been able to culture complex cultures, including immune cells, with primary tumor cells?
Yeah, so, well, I guess the definition of complex, so I think we'll probably start talking about organoids, where, we've done things like kidney, hepatic organoids as well. And the next stage where, it's a couple of customers and also collaborative studies is, where you even increase those complex structures by incorporating things like immune cells or endothelial cells, for example, and they're even growing interest in having microbial, this kind of cultures as well. So depending on what the application is, where can best advise and obviously select the right growth factors in terms of pods, just to create that complex environment that can really mimic what the customer is actually interested in, was like growing in vitro.
Great, thank you. And then Zena says, how can I be sure I choose a gel with the most appropriate mechanical properties for the cell line I'm using? Within what range is the elastic modulus of the gels?
The first question, the first answer goes back to the customer, is one to understand, the mechanical stiffness of the tissue of interest. And all this is pretty much available online, breast tissues for around 0.2 kilopascals and brain tissue. And, you can work up the scale, obviously from experience, and we do have experts that can guide you or give you that sort of information as well. So once the customer knows that, then we're in the best position to select the gel that can really, the gel with that sort of like desired mechanical stiffness. In terms of the range, in terms of our common products, we have the gels from 0.2 Pascal to about 10 kilopascal, which are like the common products we see all the time. Beyond 10 kilopascal, then you're looking at things like bioprinting based kind of applications, because that can be very stiff to handle manually. But we have technical side of things that we can advise the customer based on what they want to do. If they really want a very stiff gel.
Great. Somebody else is saying, there's two questions. Did you test the cultivation of primary fibroblasts in the PeptiGel, and does it work? And then the other one, when will more growth factors, PODS growth factors, be available? Can I create a personal growth factor panel in the near future? Interesting concept.
Okay, I'll take the first part of the question, and Natasha will take the second one. But we have to. So in terms of fibroblasts, yes, we done a range of different fibroblast culture. We have HDFs, we have the mouse 3T3, which I think that's one I was showing in the presentation. Then we've also done fibroblasts as well. So what we've seen generally, fibroblast seems to love the gel, and irrespective of the fibroblast type, and based on the way fibroblast also tends to operate, it's always nice to see how they remodel the gel to secrete ECM proteins and make it as robust as possible. And in terms of cocktail growth factors.
Yeah, so as I said before, we have a catalogue of over 90 growth factors already. And if your growth factor isn't in there, then of course we can have a discussion about whether we can make that growth factor for you. And in terms of having a panel of growth factors, yes, that is completely possible. You can put as many different pod species into your gel as you want.
Great. So I think we'll start winding up. There's a couple of final, I think, straightforward questions. Should I use pods embedded in PeptiGel or underneath PeptiGels?
You can do either. It depends on what your purpose of the experiment is. Yeah, we can have a discussion about that as well. And then
what is the source, in inverted commas, of the growth factors used?
So I'm not sure what that's referring to, but the genetic constructs that we're using are based on sequences that are from different species. So we have a range of both murine and human growth factors, and it's simply just that the sequence is inserted into the PODS crystal genetically and then expressed within the insect cell.
Great, I think that's, yeah, so they're insect derived. Somebody's also coming there insect derived, exactly. I'm going to finish off then with the last question. There are a few other questions that we will reply to, and just to let everybody know, we will be incorporating these questions into a sort of FAQ type sheet. So not only will you have the recording, but we will try and answer all the questions that we have fully on our adjacent websites. So just as a last question then, I think it was one for Dammy, and I'm not sure whether you'll be able to answer it or not, but somebody said, why the bee on the logo?
So the bee, it's a representation of the beta sheet formation. So if you think about, like so basically when the peptides come together, they self-assemble to form, beta sheet, and that's where the B aspect of the logo.
Isn't it also that Manchester, the city logo is the B?
And B as well, yeah, so it's like double B.
Double B.
So that, I don't think we can beat that actually as a final answer. That I have learned something even today. I thought I knew quite a lot about this product as well. Anyway, thank you very much, Dammy and Natasha, for presenting and answering all the questions. I'd just like to say thank you for all the people who've signed up and attended. It's been a great event, and we've really enjoyed hosting it. And as we said, there'll be a number of follow-up from the website, and we will be sending out the recording and FAQs. So yeah, on behalf of everybody, I'd just like to say thank you to Dammy and Natasha, and thank you everybody for attending, and we'll see you at the next webinar. Okay, bye.